biomedical image processing course. Today we will see the developing and defining a risk reduction program to achieve the outcome of reducing the incidence of vision loss in diabetics. Early diagnosis of diabetic retinopathy is not enough. To emphasize the objective of the program and the need to ensure timely treatment, the term risk reduction program is employed in preference to screening program. So we have to plan a mass screening and followed by fixed scheduled re-screening of the subjects. If we follow this, it will help us to early diagnosis of the diabetic subjects. To ensure its success, a risk reduction program need to be developed in cooperation with a great number of people, institutions, companies and organizations. If we go with along with all of them, a risk reduction program may meet with opposition from unexpected quarters. To try to understand why this is so, the point of view of each interested party must be taken into account. Optician and optometrists may fear for their future income and market penetration. Ophthalmologists who cherish their clinical freedom and their power over their working day might not enjoy the feeling of being told by screeners what to do and when, even if such suggestions are being made in the nearest possible way. Such reasons one has to consider while doing a screening and why people are not ready for such masses re-examinations. Economists and managers can make all sorts of objections based on need for resources and the low probability of quick monetary returns. Therefore, we have to go with by having awareness among all such entities of the society. It may be more profitable for eye clinic to perform the extensive photocoagulation program necessary for late stages of STDR than the perform early treatment that may also progression. Politicians are likely to show limited interest in intervention whose outcome may not appear as good news in time for the next election. According to the other modalities of the society, vision problems are inevitable in old age and an integral part of the disease of the diabetics. So, diabetics is come along with the symptoms which are suffering with other organs too. However, you should be able to refute such a statement. Few health economists appear to be in favor of screening programs. In the case of STDR, however, there is good paper to cite that is based on hard evidence. Be prepared to hire a health economist whose efforts could help you market your program. A good recent paper to cite comes from societies. The word screening itself has a bad name in certain areas. This may be because some people think that population-oriented intervention encourages on individual freedom. Many questions of ethics have been discussed. Such misunderstanding must have to first remove from their mind, make them aware and ready for the mass screening as well as re-examination. Other reasons that we must have to understand while doing this screening for possible criticism. Moreover, several proposed programs such as PSA screening have become the subject of considerable critical discussion, partly because their value to the individual participant is unclear. A model has been developed for use in shared decision making to show the likely consequences of one person decision whether or not to participate in such a screening program. So such mindset also need to be developed among the subject and make them aware to go for such masses screening. Some rational arguments of long standing deserve to be mentioned, at least in brief. Early enthusiasm for mass screening program based on biochemical test increased in the 1950s with advent of cost-effective automated analyzers. Good diabetic retinopathy detection program, in contrast, are based on strong evidence and should not be together with the other program of unknown values for the society, but soon warn of instance if 10 tests are performed, each with reference range covering 90% or 95% of the population. The probability of at least one false positive test come close to 50% because such attempt at screening caused more problem than were solved. Efforts were made to define why, how and when screening for disease would be acceptable. Criteria for mass screening program were published and several other criteria for screening test or program were devised later in the successive decade. It might seem unlikely that any disease, any screening test or any screening program could be acceptable according to all such strict rules. Actually, diabetic retinopathy is one of the few diseases for which screening program exists that fulfill such list of all criteria, and therefore there is consequence that a reliable list of patient, suitable equipment kept in good working order, trained photographers and graders, reference images, quality assurance scheme, etc. need to be defined in advance, as well as other factors ensuring good coverage and accurate diagnosis are necessary to get success from such mass screening and re-examination process. Setting quality assurance standard may also include a particularly useful document is called workbook. At the time of working in its 4th edition, which was published January 2007, updated in March 2008, has elaborated such a standard in detail. So one can refer this for knowing more details about the standard to assurance good quality about mass screening, describing essential elements in developing such program. The national standard are worth reading. Furthermore, Appendix 2 of the workbook, there is list of service objective and quality assurance standard has also been illustrated. These targets are very hard to meet but extremely important to get good success in these screening programs and re-examination of masses of the population. A preliminary version of those standards was tested on 10 existing services with varying results. Further information on service objective is available on the NSC website. So one can also go and explore their further details, which has provided links to other site also. In 1998, the UK National Screening Committee NSC distinguished between a screening program and screening test. By giving such differences, one can able to remove their misunderstanding about such valuable program. Now, under quality assurance standard, in 1998, UK National Screening Committee distinguished between the screening program and screening test. This program consists of all those activity from the identification of population likely to be benefited through the definitive diagnosis as well as treatment with inherent characteristics, explicit quality standard, and information system which allows comparison of performance with these standards. Managerial authority to take action to improve quality of such valuable activities. Now we will see training and the assessment process. Training courses and the criteria are in developing in many countries. See the United Kingdom website to get more details about the training and assessment program they have been followed. Under training documents and advice, the training manual is highly recommended. One has to go with such documentation to remove their misconcept about the mass screening. An Australian manual contains useful links also. An American perspective on ophthalmic photography and digital imaging of the diabetic retinopathy screening is also available on their website. Therefore, the choice training is need to be followed by all the population of society. Sensitivity is proportion to disease positive individual who are detected by the test, also known as the true positive fraction. An ideal test has an sensitivity of 100%. Specificity is the proportion of disease-free people correctly labeled as disease-free. An ideal test has specificity of 100%, meaning that the false positive fraction is 0, and therefore it is expressed as 1 minus false positive, which is ultimately equal to 1. Some efforts desired sensitivity for detection of STDR to be its least 80%, which is greater, the TPF is greater than or equal to 80%. Assessing accuracy of diagnosis test is done by certain expertise which desire sensitivity for detection of STDR type of diabetic retinopathy to be at least 80% of subject. This has reportedly been achieved by number of studies. Most of the term by high quality fundus photography through pharmacologically dilated pupils using color transparency film, trained graders and quality assurance system, because low sensitivity entail delayed detection of STDR and increased risk of vision loss. High sensitivity is important to the patient. Note that the less advanced is the disease, the lower the sensitivity will be reached to some experts desired specificity for detection of STDR ought to be at least 95%. Such way we can give the accuracy assessment, because low sensitivity entail delayed detection of STDR and which increase the risk of vision loss. High sensitivity is important to the patient. Note that less advanced is the disease, the lower sensitivity will be achieved. This has reportedly been achieved by number of studies, most of them by high quality fundus photography through the pharmacological test done on the screening process. Some experts desire sensitivity may achieve which is more than 80%, and specificity is the proportion of disease-free people correctly labeled as disease-free and which is around 100%, meaning that false positive fraction is equal to 1. Sensitivity is proportion of disease positive individual who are detected by the test, also known as true positive fraction. An ideal test has the sensitivity which is around the 100% for the subjects. By following such different methods, one can go for screening and make the people awareness that they are benefited from such type of screening. If we help them to understand that it is much more need-based and it is for their health, then they may be ready to go for such assessing method. The extreme standard for sensitivity and specificity were set during 1984 at a meeting organized by diabetics, then the British Diabetic Association, and later accepted by UK National Screening Framework. A recent review is available on this particular site. So the interested audience are kindly requested to go through this particular website to get the information about how other researchers and the scientists related to diabetic retinopathy have followed different types of methods. Most published studies have insufficient statistical power for conclusion to be made on such particular method. So if you go with this screening processes, then assessing accuracy can be improved. This criticism, however, does not apply to study in which one experience is examined during 1594. Randomly selected people with diabetics who also underwent the random methods, and their sensitivity, specificity may not be of that much accurate. So for improving this result using new method for imaging, quality assurance and grading will indeed be challenging task. Incidentally, this ophthalmologist did exceptionally well when compared to 7-field stereo photography of 2 to 9 people with the diabetic subjects, since the 1 TPF is equal to the false negative fraction. The pair TPF and FPF defines the probability with which error occurs when using such particular test mechanism. A note of caution: there are proportion of or probabilities, never call them rates in front of an epidemiologist. Moreover, the pair FPF and TPF is just one point of the receiver operating curve and thus of limited interest. Whereas if you go with other more details, joint confidence region for TPF and FPF can be presented and numbered. Rectangularly region derived from cross product of confidence interval for TPF and FPF in order to save space. As a reader, I prefer graph showing elliptical joint confidence regions based on joint asymptotic normality. This is something to look for and published papers. Many studies of diagnostics method are underpowered, which shows up as imprecise estimation, and therefore we have to see all such mechanism. Now overall conclude that while doing screening and re-examination, one has to follow a proper plan. Overall, this method if you follow, we get high accuracy. In successive point, we will go with other details also. Thank you.